Well, in Lab 22, the sheep eye dissection and the eye anatomy. So this is the sheep eye dissection part, and you need to be able to identify the basic structure in the sheep's eye. So let's look at the picture. So I put the YouTube video here. You can take a look and show you everything you need to know about the sheep eye dissection. And when you grab the sheep eye, you look like this. The front view is this. You have the cornea and sclera, so that's the outer layer of the eyeball. The cornea part is supposed to be transparent, but all the sheep eye we have, they're fixed by paraformaldehyde, and it turns it to the opaque color. So in the healthy eye, it's supposed to be transparent because the light can go through. The sclera, the white one, that's the sclera. So it's a very tough layer because it creates the structure of the eyeball. So that's what you can see from the outside, the cornea and the sclera. Then you cut the eyeball open. When you cut it open, you can cut it sagittally, you cut it coronally, both. I can explain both. So if you cut it sagittally, and the eyes have this the cornea and this is sclera. So let's look at inside. So the front view, that's the iris, and the pupil is the opening created by the iris. So and this is the lens. This is a lens, and lens was surrounded by the ciliary body. It looked like a mushroom. When we look at the other picture, it's pretty easy to see. The ciliary body is the muscle to pull the lens, so it will change the curvature of the lens, and in order to focus the light perfectly on the retina. What's the retina? This yellow piece of paper, that's the retina. So you may heard some boxers, some athletes have the retina detachment. That's because they hit the eyeball too much, and this piece of paper detach. So it turned out there's no way the light can focus perfectly on the retina. So it can cause blindness. And this area that removed the gel structure is supposed to be a gel structure called the vitreous humor. It looks like this. It's like a gel, but it's mainly water. So its function is to maintain the shape of the eyeball. And let's look at the coronal section. So coronal section, both of them show you different structures. You have the lens, and let's look at I call it the mushroom. It looks like a mushroom. The mushroom is the ciliary body, so it's pretty obvious structure. So all this ciliary body that pull the lens change the curvature. And this the vitreous humor, maintain the structure. Now let's look at the eye model. So in the lab, we have the eye model. And eye model, you need to be familiar with those 6 muscles. So outside the eye, you have 6 muscles. It looks like this. So that's the cornea, that's the sclera. So that's the top view. That's the top view. The white part is the sclera. And you can see the rectus muscle, superior rectus. Oh, and how do you identify this is right eye or left eye? You look at the cranial nerve number 2, the optic, because the optic nerve is going to go inside. So this tilt this way, you know this is the right eye. That's how I identify this right or left. And after you identify this is the right eye, you know this is the lateral rectus. This is the medial rectus. And this is superior oblique muscle. And the inferior oblique muscle, they are below. And this cranial nerve number 2. And when we take it open, okay, they show you the inside of the eyeball, and that's the retina, and this is the blind spot, also called the optic disc. The optic disc, that's where the blood vessel and nerve go in, go out. So there's no photoreceptor here, and that's why there's a blind spot. But the official name is the optic disc. And this gel, that's the vitreous humor. So all these other gel structure maintain the shape of the eyeball. And you can see this mushroom, and that's the ciliary body. So from the inside, you see the mushroom, 100% that's the ciliary body. And you say, what's the iris? Iris is a very, very beginning part. You need to look at it from the front view. We have last picture I can show you. The iris is in the front, and all the posterior part, all these are ciliary body. And this another model we have. It show you the muscles, and that's the lacrimal gland. That's your tear gland. Okay, the last one is the eye histology model. So let's look at the eye histology model. So that's the cross section. This is the cornea and the sclera. Number 2, that's the anterior chamber, and this is the posterior chamber. So what's the iris? Iris is number 30. This is the iris. So the iris gives you the eye color because the iris have the pigments. And when we look at your eye, we see this part. Now let's look at this is a lens, and this is the ciliary body. Okay, you found the whole mushroom. That's the mushroom I talk about. So from the posterior, look at the eyeball. This mushroom, the whole thing you see is ciliary body. That's a big muscle. And the iris is only this part, the anterior part. So that's the difference of these two. Then this part, that's the posterior chamber, and all inside they have the vitreous humor. And that's the fovea. That's the area give you the sharpest vision. You have a lot of cones there. And that's your optic disc. So that's your blind spot. And that's where the nerve and blood vessel go in, go out. And this is your cranial nerve number 2. So you need to be familiar with the eyeball structure, both the sheep eye dissection part and the model part. So I'm going to ask you a lot of questions from the eyeball. Okay, that's it.